Thanks for coming to my talk. I hope it lives up to this ambitious title. Starting around the 1950s, biologists became really interested in the study of clines. Clines are gradients in a measurable character or genotype. And population genetic theory that predicts clines arises from this assumption that fitness of one allele or genotype increases along an environmental gradient, while the other allele or genotype decreases. And this assumption leads to a decline in allele frequency, as shown here. As a result of this theory, statistical methods called genotype-environment associations, or GEAs, have been developed to identify clines. But we also know that neutral population structure can arise along an environmental gradient due to demographic processes, and that if we don't correct for this structure, we will get false positives when we look for clines across the genome, as shown here with these stars. And so to prevent this from happening, a number of different methods have been developed that correct for structure in this statistical test. And ideally, they separate the selected signal from the neutral one, although it doesn't really work this way in practice. These methods and empirical papers that cite them have been cited thousands of times. A scientific paradigm is a distinct set of concepts and research methods that constitute, that determine what constitutes a legitimate contribution to a field. And these methods, this GEA concept meets that definition of a scientific paradigm. But does adaptation in multivariate environments actually produce clines? A lot of these methods in theory are based on single locus, single environment models. For example, I work in oysters that have a very steep, that adapt to very steep temperature gradients across wide spatial scales, as well as gradients of salinity and disease on smaller spatial scales within estuaries. And so the overarching goal of this research is to understand under what conditions can we infer the genetic basis of adaptation in multivariate environments. And I'm going to come at this from a first principles perspective as well as a statistical perspective. The first principles issue is how genetic systems solve this problem of adapting to a multivariate optimum. So I'm going to use simulations to address this. And in the simulations, there's two environments, a temperature environment that has a latitudinal cline, and a second environment in this one has in this example that has a longitudinal cline that's shown by the thickness of the line around each population or deme. These demes are arranged on a landscape in a grid, and these gray lines show the pathways of migration. This is an estuary landscape where there's high migration within estuaries and lower migration between estuaries. The statistical issue is whether we can use multivariate statistics to infer anything accurately about multivariate adaptation. And so to address these issues, I developed some genome scale simulations with 21 linkage groups. And on half of these linkage groups, quantitative trait nucleotides, or QTNs, could evolve that have effects on traits. Those traits were adapting to a multivariate optimum on the landscape that was determined by this multivariate environment. And I simulated three landscapes, a stepping stone landscape with latitudinal and longitudinal environmental clines, a mountain landscape in which the longitudinal gradient was analogous to trees adapting to altitude or elevation, and this estuary clines landscape that might be analogous to oysters or sticklebacks that repeatedly colonize freshwater environments. These landscapes were crossed with 5 demographies with varying patterns of population size and gene flow, as shown here for a stepping stone model. 3 different levels of genetic architecture spanning from oligogenic, where 1 or 2 nucleotides would have large effects on a trait and a few smaller ones, to highly polygenic with thousands of quantitative trait nucleotides with tiny effects on the trait. And then finally, I simulated different levels of pleiotropy. So I had single trait, single environment simulations, as well as simulations with multiple traits and environments with no pleiotropy. So a mutation could only have an effect on a trait that was adapting to one environment, or with pleiotropy, in which a mutation could have an effect on multiple traits adapting to multiple environments. Those traits were under stabilizing selection within each population and I simulated different strengths of that as well. So in total, there was over 2,000 simulations. FST varied between 0.05 to 0.2, depending on how much drift there was. And there was local adaptation similar across all simulations, as evidenced by higher fitness and sympatry than allopatry. So I'm going to show you the distribution of phenotypic clines in this simulation on the x-axis. And on the y-axis, I'm going to plot the proportion of quantitative trait nucleotides that have significant clines that underlie that phenotype. So this is across all simulations. And as you can see, all the simulations involve similar phenotypic clines. But as we move lower on this y-axis, it seems that we have an increasing paradox. How are these similar clines maintained with fewer clines in the alleles that underlie those phenotypes? All of these points at the top are from oligogenic architectures. Most of them are also single trait and simulated on a simple landscape, a stepping stone landscape. As this paradox deepens, it's with multiple traits, with increasingly polygenic architectures, with mutations that have pleiotropic effects on multiple traits and more complex landscapes. So to illustrate how phenotypic clines are maintained without allelic clines, I'm going to show you an example from this area, the parameter space. So this is one of those estuary cline simulations, and I've plotted the phenotypic clines on the top. So for each individual, their trait value is plotted against the environment of the deme where they were sampled. And on the bottom, I've plotted the allele frequency patterns in the underlying alleles for those phenotypes as a function of the environment. So the x-axis is the same in both figures. And as you can see, there are some alleles that evolve clinal patterns as shown by these warm colors and these dashed lines, but there's a lot of non-linearities or what appear to be no pattern at all. So to illustrate how these patterns arise, I'm going to show you some genotype heat maps from individuals sampled at these inner estuary sites on the left and these outer estuary sites on the right. And these individuals are sorted according to the populations that they came from. So what I want you to notice here are these block patterns or modules of mutations. Then these mutations are arising that allow the populations to adapt to the multivariate optimum. So they're especially happening at these inner estuary sites in this simulation. These modules are the ones that are causing these little blips in the temperature plot. And averaged across all inner estuary sites, that pattern, these patterns cancel each other out, which make it look like there's no clines for the second environment. So this theory is great, but what about real life? How can we test for these nonlinearities? So I propose that we do genotype-phenotype associations and then look at the allele frequency patterns for those hits across environments. And I predict that we'll see some clines and some nonlinear patterns. And luckily, Colin Mahoney did this in 2019 for GWAS hits for a cold injury trait in a pine and plotted the allele frequency patterns for those hits as a function of mean annual temperature. And what I think is interesting about this paper is that I'm seeing patterns here that are eerily similar to some of the things that I'm developing in the simulation. So we need some more tests like this. So the first principles issue is whether or not clines in allele frequencies evolve. And phenotypic clines can be maintained without allelic clines under conditions that promote unique mutational solutions to a multivariate optimum. This includes more polygenic architectures that have pleiotropic effects, multiple traits with different environments, and more complex demographies like an estuary demography. So the next question is, can we infer anything about this complex adaptation from multivariate statistics? And so I'm going to focus on redundancy analysis, which is a method that seeks to identify how groups of loci covary with environments. And this method can also include a structure correction by doing a partial RDA conditional on how individuals map in their genotypes in principal component space. And so to illustrate how terrible redundancy analysis is, I'm plotting on the y-axis here false discovery rate for a univariate method that includes a structure correction. So you can see this has a lower false discovery rate. Lower is better method. But redundancy analysis and partial redundancy analysis with a structure correction both had very high false discovery rates. So they're crap at outlier detection. And to illustrate a typical example, I've plotted mutations in redundancy space. And these RDA outliers, or excuse me, these gray points are neutral loci. The quantitative trait nucleotides are colored according to their effect size. The larger ones explain more variance in the trait. And all of these false positives are these black diamonds with gray dots in the middle, all around the outside of the RDA space. So why didn't RDA work? Why did the structure correction work for univariate tests, but not a multivariate test? And what I've plotted here is how individuals now map in multivariate space, and I've colored them by their multivariate phenotype that evolved in the simulation. So what's interesting here is that RDA does seem, even though it's crap at outlier detection, it does individuals do map correctly according to their multivariate phenotype. And I can explain more why that is later. But with structure correction, it jumbles everything up. And so that's why in a multivariate context, correction for structure can make things even worse. So under what conditions can we infer the genetic basis of adaptation in multivariate environments? The converse of what's stated here is that these GEA methods are only going to detect oligogenic traits that are adapting to very simple environments and that don't have, the mutations don't have pleiotropic effects on other traits. So this suggests that what we can understand about the genetic basis of adaptation to the environment from genetic environment associations and even ordination methods is very limited. However, I'd like to end with a potential solution, and that is we don't always, maybe for many contexts, we don't really need to know the genes that are involved in adaptation. And we can still learn a lot about individuals from their multivariate phenotypes. And redundancy analysis was really good at mapping individuals into RDA space according to their multivariate phenotype. So if I back calculate the phenotype, temperature phenotype, and environment phenotype from these individuals, according to this equation, I find that RDA is very good at predicting across a wide variety of scenarios and all the architectures that I simulated the individual trait. And that was without a structure correction in the RDA. So there's obviously a lot more to be done here. I have a lot more to say, but I'm out of time. I'd like to leave you with this parting thought on the catch-22 of population genetics. And thank you for listening. And I hope my talk lived up to its ambitious title. And now I have to figure out how to stop my recording. There we go. Bye.